Hello everyone and welcome to a webinar. My name is Geraldine Hamilton. I am the President and Chief Scientific Officer at Emulate and one of the founders. It is my great pleasure today to introduce our speaker, Lorna. She's our Executive VP for European Scientific Liaison. And in her current role at Emulate, Lorna leads the Applied Biosciences Group to support services and new application development. And she also works very closely to provide scientific support to our commercial activities. Lorna comes to us with over 20 years of experience in the pharmaceutical industry, spanning from pharmacology to drug safety. And she has been a champion for the organs and chips field since the early days of the technology. Prior to joining Emulate, Lorna established and led the Center of Excellence for the technology within AstraZeneca's R&D unit. And Lorna and her team were actually one of our earliest collaborators in the development of the liver chip, back when our team was at Harvard. She also has extensive experience with multiple other chip technologies from different academic groups and different companies in the field. Earlier in her career, she served as a therapeutic area lead toxicologist for respiratory and inflammation at AstraZeneca. And today, Lorna will be providing an introduction to our platform, and then she'll be discussing data with us that highlights how our Alveolus long chip and our Airway long chip can be used for applications such as the study of viral infection and development of medical countermeasures and therapeutics. And with that, I am delighted to introduce you and pass you over to Lorna.
Thank you, Geraldine, for the kind introduction. I will just move us on and get us started. It's my pleasure to present to you the inaugural lecture, or webinar, I should say, for Emulate. The webinar today is focusing on our lung chip, and we will be looking at how the lung chip can accelerate the study of viral infection and therapeutics. So on entering this new decade, life across our planet has changed beyond all imagination. Such changes are demanding that scientists urgently adapt their ways of working to accelerate the mechanistic understanding of disease pathogenesis. Now is the time to fast-track therapies that not only treat symptoms, but also modify the course of underlying disease. Now, nature is an amazing engineer. It's created many complex structures that enable the exquisite functionality leading to life as we know it. But modelling this complex biology is extremely difficult in cell-based systems. That's where our organ chips come in. By becoming cell architects, we have created a home away from home for cells. Through a blend of design, engineering and biology, this tiny chip can recreate the smallest functional unit of an organ, enabling us to more easily and accurately study the inner workings of biology that underlie health and disease. So let me introduce you to our chip. This is a short video to help orientate you around our chip. So if we take a cross section through the chip, you can see the tall top channel here in blue and a smaller vascular channel on the bottom. These two channels are completely separate, and they're separated by this membrane, a porous flexible membrane with pores of around 7 microns in diameter. We can see cells on either side of this membrane, usually epithelial cells in the top channel and endothelial cells in the bottom channel. And with the unique feature of our vacuum pump, we can stretch where organs require it. We can flow media independently through the two channels, and if we need to, again for organs such as the lung, we can replace the media in the upper channel with air. So, why organs on chips? Well, organs on chips really help us model complex human physiology and disease. The combination of the epithelial endothelial cells create a nice tissue interface. The physical microenvironment that the chips generate through the dynamic flow and mechanical forces provide critical stimuli to cells that guide their behaviour and their functionality. We can add circulating immune cells to the chips to see cytokine and chemokine release, which is very reminiscent of clinical phenotypes. And finally, we can test therapeutics in our system from an efficacy, a safety, or a pharmacokinetic perspective by adding drugs to either side of the channel. So I'm going to show you some work that started the lung chip field off, and this is original work that was done in 2010, published in Science. And what we're looking at here is the fundamental aspects of inflammation and the immune system in the lung chip. So if we flow labeled neutrophils through the bottom channel, through the vascular channel, as you can see in the video that I've just started, they flow very, very nicely. However, upon seeing an inflammatory stimuli, we upregulate adhesion molecules such as ICAM1, and this causes the neutrophils to start to stick to the undersurface of the vascular channel. Now, not only do they start to stick, they become curious, they're motivated to move through the release of chemokines. And we observed this in this following video where we've tracked one neutrophil and we can see it weaving and wobbling around, following the chemical gradients and eventually diapodesing through the membrane to the upper channel. And if we also then look at our final video where we've now labelled our neutrophils in red, the neutrophils in this case have moved from the vascular channel to the epithelial channel in order to engulf the bacteria that was responsible for setting up the inflammatory stimuli. This is how our bodies function, and it's a unique insight into the biology. So why study viral infection, and what models do we have available to us? Well, we have many models available to us, from culture cell lines, precision cut lung slices, lung organoids through to our organ chips. Now we know that each system, each model isn't perfect. No model's perfect. However, there are significant disadvantages for culture cell lines, precision cut lung slices, and indeed lung organoids that actually prevent it from completely modelling the complex biology. And this is where the organ chips come in. I'm going to show you data in our presentation that will hopefully illustrate why we can actually start to recreate physical and clinical phenotypes and responses at clinically relevant drug exposures. But we also have animal models available to us, and many people use animal models particularly to study respiratory viral infections. But it's becoming increasingly clear that there's many species differences when it comes to viral susceptibility. And actually, when we look at the translation of inflammatory responses in animals to humans, they don't always correlate. So as a consequence, we see a very limited recapitulation of the clinical phenotype. This means that people often migrate towards non-human primates, for instance, and that leads to expensive and complex husbandry requirements. So our thesis is that organ chips can actually help provide an ideal model system to study respiratory viral infection through the use of human cells on these chips. So I'm going to start by introducing the Airway lung chip to you. So first of all, I'd like to show you a video which nicely illustrates the characteristic pseudostratified epithelium. So that's the elongated epithelial cells with the densely packed nuclei. And this is particularly characteristic of the human airway that you would see inside the body. Below, you can see the endothelial cells forming a nice tight monolayer. And if we go and look at those cells in a bit more detail and start to try and characterize them, what we can look for are the main cells that we would expect to see following around about 14 days of differentiation under an air-liquid interface. So starting on the left-hand side, the first image that you can see is of club cells. And here we're using a marker, CC16, which is a secreted protein from club cells, and they are stained in green. The next image along is the ciliated cells, and here we're stained for a major protein in the ciliated cells, beta-tubulin, and that is stained in red. And finally, when we look for goblet cells, here we've used the stain for MUC5AC in green, which is one of the major mucins produced by the goblet cells. So overall, our chip has the correct phenotype of cells that are all fully differentiated when cultured across an air-liquid interface. And to show you this in a little bit more detail, this is a beautiful scanning electron micrograph of the cilia on our chip. The ciliated cells have been stained on blue. The non-ciliated cells are the brown cells, which are going to be a mixture of goblet cells and club cells. And to show you this in a bit more detail, to show that not only do they look nice, they actually do function. So the video that you can see on the left-hand side is the cilia beating nicely. And the video on the right-hand side is a blob of mucus that has been secreted from the goblet cells and is actually moving as a consequence of these cilia beating. And one final demonstration of the functionality here of these ciliated cells, if we were to place fluorescent particles into the airway channel of our chip, we would observe this characteristic upwards movement, which is where the cilia are trying to sweep these particles away, and if it were in the body, away and upwards, and they're ultimately out of the body. So I hopefully have shown you that the airway lung chip has human relevant biology, and here's some further facts and figures that we can demonstrate that principle. But what I really want to draw your attention to are the two figures at the bottom of each of the circles. On the left-hand side, it's the human lung, and on the right-hand side, it's the human lung chip. So I've demonstrated that we have the key cell types that are present, but actually what's also important is the ratio of those cells. So in the human lung, for instance, around about 30% of the cells are ciliated cells. We see in our lung chip 20 to 30% of the cells that are ciliated. And equally, if we look for goblet cells, the human lung, 10 to 15%, whereas in the human lung chip, we're somewhere in the region of 10 to 20%. So we believe we've really recreated the lung airway. So let me now show you how our airway lung chip responds to human rhinovirus infection. So human rhinovirus we used here with type 16, so I may refer to it going forward as HRV16. And on the graph on the left-hand side of the slide, what you're looking at is the viral titer on the y-axis against the time post-infection on the x-axis. So you can see within the first 24 hours, there's a nice increase in the viral titer in the chip, indicating that the virus is able to replicate inside the chip. It remains steady for a further 24 hours and then gradually declines over the course of the experiment. Now, if we were to look at the impact of the viral infection on the epithelial cells under a bright field microscope, you would see the images on the right-hand side of our slide. The first image is of a non-infected cell, where if you look very closely, you can see the nice epithelial cell structure. But on the right-hand side, what you will see is a chip that has had HRV 24 hours post-inoculation. And what you can now see is you can see that the epithelial cells are very rounded, and that's indicative of very extensive epithelial damage. So we decided to have a closer look at what was happening with the HRV infection. So it's been reported in the literature that the ciliated cell is actually a key target for human rhinovirus. So here we have our ciliated cells on the image on the left-hand side. This time beta-tubulin is stained in green. And what we're looking for here is double-stranded viral RNA, which indicates that the virus is actually replicating, and we've stained for that in red. So these ciliated cells are clearly packed full of this double-stranded RNA. Now, if we were to also take chips that had been exposed to human rhinovirus and use a tunnel stain, what we can actually see is a large number of green cells in the middle image, and that's indicative of apoptosis. And indeed, it's been reported that the HRV infection causes a sloughing and apoptosis of ciliated cells. So not only can we see this morphologically within the lung chip, we can actually measure the functional consequence of that. And that's the graph on the right-hand side of the slide. Here, you're looking at cilia beat frequency, and in the open bar, that's the control chips or the non-infected chips. And you can see in the grey bar, around about a 50% reduction in cilia beat frequency because of the HRV infection. Turning our attention then to the goblet cells, we also know that HRV infection causes a hyperplasia within the goblet cells, and that's to increase the amount of mucus that the cells produce as another line of defense to this viral infection. So looking at the image on the left-hand side, the non-infected cells, you can see some red staining for the goblet cells, which is exactly in line with what we would expect. But 24 hours after HRV infection, we can actually see a large increase in the number of goblet cells, representing this hyperplasia that's been reported. And the graph on the right-hand side shows you the quantification of the change from the non-infected chip to the infected chip in a 24-hour period. Now, clinically, we know that rhinovirus infection will cause the release of cytokines, both that are pro-inflammatory, as well as chemokines, to also bring in another cell type into the airway. So here what we're looking at is CXCL10. It's one of the members of the interferon family, and again, it's clinically been demonstrated that the interferon response is a very early and necessary response to fighting the rhinovirus infection. So you can see that the chips infected with HRV in grey show an increased amount of CXCL10. The next two graphs, the first one MIP1 alpha and the one on the far right, MCP1, also show increased levels of secretion following viral infection. And these two chemokines are known to bring in the alveolar macrophages, again as another line of defense to fight the infection. So let's look at the airway lung chip now with a different virus, and this time we're looking at influenza virus. This work was actually done with our partners and collaborators at the Wyss Institute in Harvard. And what we're looking at here is, and again, some nice immunofluorescent images of the epithelium, but additionally this time the endothelium. So starting on the left-hand side, the upper panel is the control, and here we're staining in red for a tight junctional protein called SO1. And what we know about influenza infection is that it likes to break down SO1 and break down these tight junctions. So if you look at the image below, which are chips that have been exposed to the virus, you will see that there's an absence of SO1 staining. Similarly to the HRV infection, influenza also affects the ciliated cells. So the next image that you can see are cilia stained in yellow in the control chip, but in the presence of virus, again, a real loss of ciliated cells. Now this time the virus was a GFP labelled virus, so we were able to detect it in green. In the upper panel you can see no virus, in the lower panel you can see that the virus has actually infected cells. Now additionally we're looking at the endothelium, so the underside, the lower channel of our chip. And for the control here we've stained for VE cadherin, again to show that we have nice tight junctions in the endothelium. But the presence of the virus, which was only added to the epithelial layer, has actually made its way through to damage the endothelial layer, and this we believe is responsible for the vascular leakage that's often reported in influenza infection. So what happens when you start to try to therapeutically intervene in the airway, when the airway lung chip, when we're infecting with virus? So in this experiment, oseltamivir acid, OSV, was used, which is the active ingredient of Tamiflu, which many of you will have heard of before. Now, Tamiflu is used clinically and is known to be particularly effective if it's given within the first 48 hours of infection or first 48 hours of a patient reporting symptoms. So here, the data we're looking at is all 48 hours post-infection, and starting on the left-hand side, what we're looking at is the relative titer, so the amount of virus that's in the chip, and we've normalised it to the control. And you can see that with 1 micromolar of OSV, we can bring the viral replication right down by about 90%, so very, very significant impact. Now, remember we said that the virus actually breaks down the tight junction, and when that happens, the barrier that's there becomes leaky, and we can measure that using the apparent permeability. So in the presence of virus, which you see designated on the x-axis as a plus, there is an increase in apparent permeability indicating a leaky barrier. However, when virus and OSV are added together, that tight barrier is actually reformed. And we can see this morphologically by the images on the right-hand side. And these are actually some of my favourite images. So we're looking first of all at the control, where ZO1 is stained in red, really nice tight junctions. Secondly, the virus taking away those tight junctions. And then finally, the restoration of those tight junctions with the addition of the oseltamivir acid. So this explains some of the therapeutic benefit that oseltamivir acid will actually generate. But we also know that chemokines and cytokines are an important response to viral infection. And so here we've looked at chemokines and cytokines that are known to be part of the clinical phenotype. And again, 48 hours post-exposure, the chips that received virus plus OSV show very much reduced cytokine and chemokine release, again, contributing to the therapeutic action of this drug. Now let's turn our attention to a slightly different drug. This time it's nifamostat. Now nifamostat is actually a clinically used anticoagulant, and it's very effective as an anticoagulant because it's a particularly potent serine protease inhibitor. And what we know about viruses is they like to gain entry into the cell, and they rely on serine proteases to help them do so. So here, when we give 10 micromolar of nifamostat, again, the data, here we're looking at is 48 hours post-viral inoculation. First of all, we can look at the barrier integrity. So again, here using the apparent permeability, empty chips clearly would be very leaky, so we see quite a high apparent permeability. In the presence of the virus, but when cells are there too, we equally see a very high permeability. But interestingly, treatment with nifamostat over a 48-hour period reduces that barrier leakiness, restoring that barrier integrity. And again, morphologically, we can see this with the restoration of the tight junctions by the reappearance of the ZO1 protein in the nifamostat-treated chips. And similarly to the picture for oseltamivir acid, we're looking here at the chemokines and the cytokines that are produced following viral infection. And we can see very clear evidence that the inhibition of this host serine proteases is leading to a reduction in inflammatory cytokine and chemokine release. So part of the therapeutic benefit that this drug will give to influenza patients is clearly around the serine protease inhibition and reducing the amount of viral entry into the cell. Now, one of the reasons I love organ chips so much is that we can actually do combination drug testing in the organ chips as well. And it's very clear for viruses such as the SARS-CoV-2 virus that to be successful in therapeutic treatment, we will more than likely need more than one drug. So combination drug testing will be what we need to do to see therapeutic benefit. So in this graph, we're looking at two different scenarios. First of all, at the minus 24-hour time point, we're looking at a prophylactic treatment. So that means that in this occasion, drug was given to the chips before the virus was given. Now, this is a very artificial situation because I don't know of anybody who goes to the doctor 24 hours ahead of time and say, I think I'm going to have a virus tomorrow, please can I have antiviral drugs? That doesn't happen, but it's actually a really good test just to make sure in a complex cell system, for instance, that you can add virus, as we have done to the control chips in white, you can measure the viral titer. But then when you add the drugs either independently or together, prior to the virus, of course, you can see that there's very much a reduction in the viral titer. But what's more realistic is what we call a therapeutic exposure, and that's when drug is given at the same time as the virus onset. And if we follow that course of time from 0 hours through to 96 hours, we can look at how each drug works itself or in combination. So the light grey bars are the nifamostat by itself, and the dark grey bar is the OSV by itself. And you can see that both drugs will reduce the viral titer out to about 48 hours. And this is definitely in line with what was being reported clinically. But what's really interesting is that if you give both of the drugs together in the black bars, we can actually keep that viral titer down out to 96 hours. So in other words, we've extended the therapeutic window for a further 48 hours. Now I'm going to change gears and talk about the alveolus lung chip. You'll recall at the beginning of the presentation, I highlighted that organ chips are actually representing a fundamental functional unit within the lung. And the alveolus is an independent functional unit of the lung as opposed to the airway. It has its own unique structure, microenvironment, and of course functionality. So we've adapted our protocols and used the adapted protocols with alveolus cells to build an alveolus lung chip. So in this occasion, we've taken human primary alveolar epithelial cells. We've seeded them again on the upper channel of our chip. And looking at the image on the top right-hand side, you can see beautiful zone 1 staining in green, showing very nice tight junction monolayer formation, which is very typical of the human alveolar epithelium. We've used microvascular long endothelial cells on the bottom channel, and again, looking at the images on the right-hand side, you can see we've stained for VE caterin in pink, showing again very nice intact tight junctions. And just to give ourselves confidence that we're looking at the right endothelial cells, we've equally stained for von Willebrand factor in green. So far, all of this data has been unpublished. Now, those of you who are working with alveolar cells will know that actually it's incredibly difficult to keep the type 2 pneumocyte in a type 2 phenotype once it's removed from the body, because it rapidly wants to de-differentiate into a type 1. And what's very exciting about the images that you're looking at on the screen now is that the unique alveolar microenvironment that we've created has enabled us to keep type 2 cells through at this point in the slide out to day 7 under an air-liquid interface. And how do we know that? Well, in the middle image, you can see that we've stained in green for prosurfactant C, which is one of the putative markers for type 2 cells, because their function, of course, well, one of their functions is to secrete surfactants. Another putative type 2 marker is HT2-280, and so the image on the right-hand side, you can see we've stained for those cells in green. So we can see type 2 cells. But the image on the right-hand side, we're also staining for podoplanum. It's a type 1 pneumocyte marker. So you can see that in our chips at day 7 under an air-liquid interface, we actually have the correct microenvironment that will support both type 1 and type 2 pneumocytes. And if we look at the gene expression, What we're looking at here on the left-hand side are surfactant C and B to represent the type 2 pneumocytes, and on the right-hand side, aquaporin 5 and podoplanin for type 1 pneumocytes. And we've compared the gene expression in chips to static 2D cultures. So you can see quite clearly that surfactant is present in both the chips through to actually day 15. And we similarly see our type 1 markers also present in the chip out to day 15. Now, the alveolus, in the process of breathing, it curls and uncurls, and that's partly how the lung becomes a very efficient gas exchange organ. So the image, the movie, I beg your pardon, that you're looking at on the left-hand side, if you look very closely, you'll actually see these cells moving. And this is due to the unique ability of our system to provide a mechanical stretch to the cells. Now, we were concerned that when we add the mechanical stretch, or indeed that we start to add the air-liquid interface, that this may affect the tightness of our epithelial barrier. So the graph on the right-hand side shows again the apparent permeability that we're measuring, first of all, very high in an empty chip where there's no cells. But you can see that we maintain a very tight barrier in this graph out to 9 days, either under the liquid-liquid interface or the air-liquid interface, and with mechanical stretching throughout. So this mechanical stretching doesn't actually disrupt that monolayer integrity. It's very important to show that we have the physiology there. Now I'm going to show you how we've used our alveolus chip, this time for risk assessment, and this was done in collaboration with one of our pharma partners. And what they asked us to do was they asked us to try and recreate an in vivo response that they had seen in their animal studies with one of their drugs. So in this particular experiment, PBMCs were added to the alveolar channel that had been pre-incubated with drug. And what they learned from their animal study was that the drug target was present on the alveolar cells, on the epithelial cells. And they wondered if we could also see a similar effect that they saw in the animals. So we added the PBMCs that had been incubated with drug into the alveolar channel. They did indeed engage with their drug target present on the epithelial cells. This caused the cells to become, the PBMCs to become activated and start to release cytokines. This cytokine release continued to ultimately the point where you saw apoptosis of the cells, and ultimately damage was reported. So in any good drug study, we started off firstly by looking for the presence of the drug target in our cells. So at day 3 under a liquid-liquid interface, day 6 and day 8 under an air-liquid interface, we stained for the drug target in pink, and you can see that it is present throughout the experiment. So what we did was we added drug on day 8 and followed the course of events for a further 48 hours. And what we saw was immune cell toxicity 48 hours after drug exposure. So if you look at the top images, these are bright field images, and the bottom images, and the ones with the blue cells, are live imaging. You can see then for the vehicle cells, if you look very closely, you can see that beautiful alveolar epithelium. And then if you look at the live cell imaging below, you can't see any PBMCs there, nor can you see any apoptotic cells, which would be in pink. Now, if we gave the cells some PBMCs that had a negative control in it, so not the drug of interest, the bright field image, immediately you can see the PBMCs as the lighter, brighter circular cells in the bright field image, and down below the lighter blue cells with the occasional pink apoptotic cell. But really interestingly, when we gave the PBMCs that had been pre-incubated with the drug, you can clearly see that the epithelium has started to become damaged. And that we can back up with the live cell imaging with the increase in the number of pink cells, indicating a larger number of apoptotic cells. Now, in the animal model, this was accompanied by a cytokine response. So using clinically relevant concentrations of drug, we also look to see whether we could see in the chip the cytokine profile that was observed in the animal models. And these graphs show you across 4 different cytokines that there was a concentration and time-dependent response of the drug to the cytokine release. So this shows us that the chip can recreate the immune cell interaction and cytokine response, and this contributes to the overall risk assessment that the company wanted to perform. So I'm going to finish the presentation with a few slides on infectious disease applications and focus particularly on the topic of the moment, COVID-19. So organ chips are perfectly positioned to help in the research and understanding of COVID-19. So I'm going to show you some data in a minute around ACE2 and serine protease TMPRSS2 expression in our lung chip. But we also know that these 2 key proteins are also present in our intestine chip and in our kidney chip. And these 2 proteins are extremely important for the attachment and entry of the virus into the cell. Our partners at Harvard have actually shown with a SARS-CoV-2 pseudovirus that they can actually infect our lung chips with the virus. So clearly, they can attach and the virus can enter the cell. And I've just taken you through some data, albeit with different viruses, HRV16 and influenza virus, that these viruses will replicate inside the cells, ultimately be released from the cells and cause damage, such as barrier integrity and cytokine release. So the lung chips are perfectly positioned to look at potential therapeutic strategies against viral attachment, viral entry, viral replication, and subsequent inflammatory cytokine storms that occur when the cells burst open. So looking then at the airway lung chip and the expression of proteins that we know are important for the SARS-CoV-2 infection. The graph on the left-hand side is looking at serine protease expression, and it's comparing the increase in messenger RNA expression in fully differentiated chips compared to in black MDCK cells, which are cells cultured in a dish in 2 dimensions, or to the grey, which is the cells on the chip that are undifferentiated. So we can see that 4 key serine proteases are all increased in differentiated chips compared to the cell line or undifferentiated chips, making this a good system for studying serine protease expression and the effects and functionality that that brings. On the right-hand side, we've stained for angiotensin converting enzyme receptor 2, or ACE2. You can see that undifferentiated chips, there is an absence of ACE2 expression because there's no green staining, and we stained ACE2 in green, whereas the differentiated cells on the right-hand image show that the ACE2 is clearly present once the cells have differentiated on the chip. So this gives us key information that proves our lung chip will be good for studying viral attachment and cell entry. Now, although this virus is very new, the amount of research that's going on is actually quite phenomenal. And if you follow some of the literature, what you'll also begin to understand is that this virus has different pathogenesis in different organs. And at Emulate, we have intestine, duodenum, and colon chips. And we've already demonstrated that we know that the ACE2 is actually present within those chips. So our chips would also help in the discovery and understanding of how this virus is actually operating. Also, clinically, many very severe patients come into the hospital and they present with altered liver function. So we have a beautiful liver chip that will also help us understand how the virus impacts the liver. And finally, it's become clear in recent weeks that ACE2 is also present on endothelial cells. And if you recall, it's one of the unique features of our organ chip that they have the endothelial cell present. And at Emulate, we've spent some time developing a blood vessel chip to allow us to look at thrombosis. And it's become clear that some patients with COVID-19 actually have an increase in the number of thrombolytic events. And we believe that our blood vessel chip would be ideally suited to help us study this too. So in summary, organs and chips for COVID-19 research, we can think of it in 3 different ways. First of all, we could consider the chip and its value in basic research, looking at pathogenesis in multiple organs, hypothesis testing from computational models and artificial intelligence. And finally, and very importantly, we believe that they can stay for the long term in terms of doing retrospective analysis to help prepare for future outbreak responses. We can also use them in drug discovery and development, where we can test the efficacy and safety of potential therapeutic candidates and accelerate the repurposing of drugs that's currently underway. And finally, we also believe they have a clinical application through the discovery and validation of biomarkers, as well as being able to explore potential variations in patient populations. And so with that, I'd like to close this webinar, and I'm very happy to take questions, which I hope you've all been doing through the chat function as I've been talking. Thank you very much for your attention.
Thank you, Lorna. As Lorna just mentioned, we're now going to use the rest of our time for the hour for a live Q&A session. We received a lot of great questions during the presentation, so we're going to start with those. But as a reminder, if you have any questions, just click that question mark icon on the right-hand side of your screen, submit your question, and we'll add it to the queue. Our first question is about the cell types used in these studies. So what epithelial and endothelial cells are used in the alveolus and the airway lung chips?
Yep, that's a really good question. So all of the data that I've shown you today have all been from human cells. So we procure human cells from the airway and for the alveolus, as well as the endothelial cells from commercial suppliers. So that from the airway perspective, we use airway cells, as well as they will have some basal cells, which we start to mature through factors in the media. And the alveolus cells, as I say, are directly isolated and contain type 1 and type 2 cells.
And to follow up on that question, are the endothelial cells that are used lung specific?
Yes, they are. We always do that when we build our organ chips. We always use endothelial cells from the organ of interest, because there are actually a number of differences in endothelial cells from different organs. So to have the right organ chip and have them, let's say, have an alveolus chip, it wouldn't make sense to put endothelial cells from the liver in there, for instance. So yeah, the endothelial cells are also pulmonary in nature.
Our next question is about the length of cell culture. How long can the cells on the chip be in culture? This is important for repeated exposure of test articles. And can this be used for testing COVID-19 infection, assuming they have ACE2 receptors? So 2 parts to that question.
Yep, absolutely. So let me start off, first of all, with the alveolus chip. We would deem our chips to be mature from 8 days, and we are confident that we have good viability out to 12 to 14 days. So maybe a period of 4 to 6 days for the alveolus chip. For the airway chip, it takes us around 2 to 3 weeks to mature the cells. But from that point onwards, we can have at least a further 2 to 3 weeks of good viability of the cells. To the question then on whether that would allow treatment for therapeutics, absolutely. of course, it really depends upon the endpoint and the duration of action that the individual would like to study. But I believe there should be sufficient window there in both of the two chips to assess the therapeutic effects. To the second part of the question around ACE2 expression, I showed some data that we can find the ACE2 in the airway of the airway chip, the cells that are there, once they're fully differentiated, so out to any time from about 2 to 3 weeks after seeding onto the chip. At this moment in time, we haven't actually yet stained our alveolus cells for ACE2 receptors. However, this is work that we will shortly be doing at Emulate.
Our next question is about imaging. So how do we label the epithelial and endothelial cells for fluorescent microscopy? Is it possible to label them while the cells are still inside of the organ chip and then examine them under a confocal, or does this require taking the cells out of the chip?
Yep, that's a really good question, and one which I really like to answer. So yes, you can do it in either way is the short answer. So most of the imaging that you saw, with the exception of a few images in the alveolus chip, was done as what I would define as a terminal endpoint. So that's where at the end of the experiment, we fix the cells in the chip and we apply our immunofluorescent antibodies and then look at the cells under a confocal microscopy technique. But you can apply cell trackers to look at live cell imaging, and that's what we did in the alveolus chip. And we know that we can do this fairly well. We can take the chip off the fluidic platform for about 30 minutes. The flow will still remain. So perfectly good time window to actually do some live cell imaging. I'm less of a fan of live cell imaging, if I'm being very honest, because I think there can be some challenges with phototoxicity. So it does require perhaps further optimization than a terminal immunofluorescent endpoint would require.
We have another question on endpoint measurement. So can you recover cells after their culture in the lung chips? And is there enough biological material to perform techniques such as PCR, WB, or ELISA?
Yes, absolutely. So in terms of ELISA, most of our ELISA measurements are done from the effluent of the chips. But yes, we can take the cells at the end of an experiment. We can gently tryptanize the cells off the chip, and there is sufficient material there to do PCR, and we can actually also do Western blotting if that were appropriate as well. So yes.
A follow-up question on imaging techniques. I think you started to mention this before, but what microscope setup do you need in order to do live cell imaging?
Yep, so we just use a standard laboratory microscope that has the correct lenses and bulbs that enable us to do it. So nothing out of the ordinary, nothing different to what would be in a standard laboratory.
Our next question is there potential to include the lung chip in studies for vaccine development?
Yes, so I believe there is. I haven't seen yet any data on any chip, actually, whether that be ours or any other chip on the marketplace at the moment. But it would require a little bit of further development, because clearly one of the roles for vaccinations and proving efficacy of vaccinations is being able to see the appropriate immune response. So we would more than likely have to adapt our current protocols to allow further inclusion of immune cells in the chip. And once we were confident we had that, then yes, we could use them for vaccine evaluation.
Our next question is about using chips as a translational model. So at Emulate, do you get requests from clients for animal organ chips as sort of a halfway house before going into human models, and to support confidence in chip models based on their ability to recapitulate animal data that is more familiar to end users?
Love that question. Very, very close to my heart. Absolutely. So actually, AstraZeneca, Janssen and Emulate published a paper that made the cover of Science and Translational Medicine in November last year, and that's exactly what we did. This was in the liver chip. We created a rat, a dog and a human version, and the idea behind doing that was exactly to address the question of translation. So that paper nicely outlines how we were able to recapitulate responses that had been seen in vivo in rats or in dogs in the liver chip, and moreover show in one of the cases that had we actually not stopped a molecule because of a response in a rat and taken it forward to the clinic, the data from the chip would indicate that actually the human had no response. So perhaps we didn't have been as sensitive to the response that the rat was. So that really opens the question about how many drugs don't make their way through because they show positives in animals. So I'm a massive fan of being able to do this across more of our models. I would love to have the opportunity to do that in the lung models, the intestine models, and indeed any of our other models that we currently have. So very much welcome people coming forward and talking to me more about that, because I do think it will help build confidence that if we can continually reproduce effects from animal models in our chips and show differential responsiveness to humans, it really will open up the opportunity to discover new therapeutics.
Our next question is directed at Geraldine. So can you expand more on the types of endpoints that you can measure on the chip? Can I measure what I would normally measure in conventional cell cultures or animal models?
Yes, indeed. I think that's a great question. I like that question because I think it's something a lot of people, when they first see the system, wonder about. And I think basically the answer is anything you can measure in conventional cell culture or an animal system, you can measure in the chip, from RNA, gene expression levels, transcriptomic analysis, anything that's eluded in the chip, in the supernatant that flows out of the chip, cytokines. In the liver chip, for example, we measure LDH or other secreted enzymes. So I think the answer is if you can measure it in your regular normal studies in conventional culture and in animal studies, for the most part, you can measure it in the chip. Now, some endpoints require optimization of the assays, but that's usually more related to finding the right kit and not so much to do with the chip.
Thank you, Geraldine. Our next question is on comparison to conventional cell culture. So how do organs on chips compare to standard 3D cell cultures? And for this question, if you'd be able to answer both for the lung chips that you described today and just on a larger note that we see in other organs as well.
Yeah, so and again, another really fantastic question, and we ask this question a lot, and for those people who know me well, know that I always boil it down to the why bother question, because yeah, why bother with these chips? So let's just think primarily here about viral infection, because that's been the main topic of the presentation. Cells in culture can be easily infected by virus. So I think there's lots of data out there in Vero cells and the MTCK cells or WHO cells, for instance, where the virus has actually been shown to infect the cells very easily. But what's not often apparent is that many of these cells, so Vero cells, for instance, are actually interferon deficient. So to actually get the viral infection and to start to model the responses in that cellular system, you actually have to add the exogenous interferon into the system to get the cells to work in the right way for your experiment. Some other cell types also lack things like serine proteases, so they have to be added in an exogenous way. And quite frankly, one of the reasons why the cells in the culture perhaps have a disadvantage is that they sit in a very static environment. In other words, they're completely submerged in media, and they sit in that media for 24, 48, sometimes even up to 96 hours. So again, not really representing what you would see in the body. On the flip side, with the chip, where we see the advantage is that the cells have this dynamic fluid flow, which means that in its simplest terms, it delivers nutrients, removes waste from the cells, and this increases their viability. But that also gives shear stress, which is a key stimulus to the cells. And together with that and the care around the microenvironment that we take when we build the lung chip, so I showed for the alveolus that we actually apply a stretch because that's what these cells see when they're inside the body. Those also give the cells additional stimuli. And those all together, what that means is that the cells not only are happy, they are viable for longer, but they actually function in a way that they would do in vivo. So for me, there's a clear advantage in using the chip. And I think the second part of the question was whether we see similar advantages in other organs. And one of the other organ chips that really springs to mind for me is the proximal tubule. So here, one of the main jobs, as people will probably appreciate, of the proximal tubule is around reabsorption of key nutrients prior to the excretion of the fluid from the body. And what we know is there's many drug transporters are present on these epithelial cells. And in a 2D culture, you can actually find the messenger RNA for these drug transporters. But because the cell is not polarized correctly, the transporter doesn't sit either in the apical or basolateral membrane, and as a consequence doesn't function correctly. So when we apply our chip technology, we can actually see the correct polarization of the proximal tubule epithelial cell, and as a consequence, we can actually look in great detail at drug transporter activity. Now, for those people who are interested, we had a poster at the virtual Society of Toxicology meeting where we demonstrated good functional characterization of these drug transporters in the chip. So different model and another example of how the chip and the microenvironment is really important to enable us to drive the right functionality.
Thank you, Lorna. So we just got the 1 minute notice here from GoToWebinar. So unfortunately, that's all the time we have for the live Q&A. I want to thank everyone for joining today's session. If you asked a question and we were unable to answer, we're going to be following up with you directly. And if you have any additional questions or just want to learn more about the lung chip, please email us at info@emulatebio.com. You can also check out our lung chip page on the website, and we're going to be adding some more links there, including links to the publications whose data was shared today. Thank you, and have a great day.